A lot of people picture scientists at a lab bench with vials full of liquids, mixing them around, or simulating events on a computer. But fieldwork is an important part of science. This week's story is about a field researcher whose contributions to virology resulted in a novel discovery. This is about the virus the cat dragged in. This is Pepper. He does local small animal surveys in Gainesville, Florida, and occasionally brings samples of interest to his colleague, John Lednicki, a virologist at the University of Florida who hosts Pepper as a scholar in residence. A few years ago, Dr. Lednicki and Pepper began to collaborate more closely. That is to say, in 2021, Pepper brought Dr. Lednicki a sample of Paramiscus gossipinus mouse. At the time, the Lednicki research group was investigating whether wild mice are vectors for a particular kind of virus, mule deerpox virus. Pepper's sample from the field provided an opportunity to investigate that very question. It turns out they found something more interesting than that. They found a new viral species and the first example of its genus, the Geelong virus, in the United States. The discovery was published last year in the journal Pathogens. But Pepper is not a scholar who rests on his laurels, although he does frequently rest in warm rays of sunlight. Pepper has continued bringing potential samples of interest to Lednicki, who recognizes Pepper's valuable contribution, and, in a new paper, Pepper has done it again. Samples from a shrew obtained in the field have yielded yet another new virus, this time a mammalian orthoreovirus. Once again, Pepper is at the forefront of virology. Of course, most have described this story as a cat caught prey and brought it home, and as humans a virologist who chose to sequence the samples because that's his specialty, and he applied his scientific background to derive scientific value from the animals rather than simply disposing of what his cat had hunted. And admittedly, there are no indications that Pepper has a specific skill for identifying novel viruses. There's no evidence this is a scenario like dogs that can detect incredibly small quantities of volatile organic compounds, where Pepper could somehow suss out a viral genome and know it's different than closely related viruses. But there are two reasons one might find a novel virus. The virus itself really is new, or it's been there for a long time and we simply haven't detected it. Like, for instance, if it doesn't usually cause disease. Like the mammalian orthoreovirus family. Which is one of the reasons, I'm going to say it right now, Pepper has done brilliantly throughout his fieldwork and has never been sick or shown any symptoms as a result of his sample collection. Nor have his collaborators because, of course, they're virologists and have plenty of equipment and protocols to study things safely. Thousands of cats, like Pepper, engage in this kind of fieldwork and sample collection every day. The only difference is that not all of them have access to collaborators who will run viral culture on those samples. In fact, culturing and sequencing the virus is the whole of this paper. It's pretty rare to come across a 4-page biology paper like this, but the story goes: sample acquired and processed, sample cultured in the dish, sample sequenced, and voila! Here's the genome of a novel virus we found. It's categorized in the journal as an announcement, in part because it's not the kind of paper that's all about testing a hypothesis. These virologists wanted to see if there was anything interesting in the shrew sample. They found something. Boom! Article. It's not to say there's nothing of interest here to non-virologists. I, for one, didn't know much about how one cultures viruses. As a reminder, viruses don't work like living cells. They have to enter a living cell and trick it into using its protein-making machinery to make more virus, then leaves the host to continue its cycle. Some viruses are more and less fancy. Some have sequences that add to their stealth, or retroviruses integrate directly into the host cell's genome. There are all sorts of bonuses, but the important thing is that the virus does not have more ways of making itself. So, when you culture a virus, it's not just like bacteria. You can throw bacteria like E. coli in a dish, and those suckers will just slurp whatever nutrients you threw in the petri dish, and the bacteria will start replicating. You throw a virus in a petri dish, and nothing will happen. And eventually it'll fall apart through pure chemical instability. If you want to make more virus, like, for instance, if you want to sequence the virus to find out what you're dealing with, you will need cells to host them. But here's the problem. Many viruses are bad for cells. If a cell is using its energy to make virus, it's not using that energy to make the stuff the cells actually need. So, we as animals have mechanisms to detect likely viral interference, and a ton of our immune system is about trying to detect that virus and then eliminating it. For example, in humans, we have a whole mechanism that pops off the moment our cells sense any DNA floating around in the cytosol of the cell, rather than tucked away in the nucleus where our DNA is supposed to be. See, DNA floating around in the cytoplasm could be from a virus that has just gotten inside the cell. So, if our cell detects DNA outside the nucleus, sure, maybe there could have been some innocent explanation. But it could be a virus, and our standard operating procedure is the biological equivalent of nuke it from orbit. It's the only way to be sure. All of which is to say, virologists use special cells when culturing viruses because most cells would not allow for viral replication without a fight. One cell type used in this paper is a monkey kidney cell that doesn't produce interferon, which is the, hey guys, I think I found a virus signal. And another cell type is from bat lungs. Bat have a highly unusual interferon system with a bit more of a I won't bother you, you won't bother me relationship to viruses compared to most species. It's one of the reasons bats tend to harbor viruses, because they can, without their immune system going totally aggro. As you may have heard in a public health context, that means bats can carry a lot of viruses that are way more dangerous to us than they are to the bats. But for this science, where you're actively trying to culture a virus, bat cells work to our advantage. For this paper, of course, none of the viral culture is terribly innovative. It's all pretty run-of-the-mill in virology. But for those of us who don't know a lot about how scientists actually work with viruses, it's cool to learn a little more about how they do what they do and all the clever things, like using bat cells, that they do to allow for that. And even though we are not bats, and our immune systems do pop off at the sight of viral infections, it's important to remember that viruses, like bacteria, can be part of our bodies, and it's totally normal for the animals to have viruses inside of us. Just like some bacteria can make you really sick, it's still normal to also have other gut bacteria, and you to die without them. So when Pepper found that mouse, and later that shrew, it's likely that the viruses those animals were carrying were just viruses those animals happen to be reservoirs for. That means it's unlikely that Pepper brought back these field samples because he could somehow detect there was anything unusual to them. There probably isn't. And even if there were, Pepper is a field researcher. He doesn't necessarily access the viral genome database to know what has been published about. The difference is that this time, our field researcher was paired with a virologist who actually took a look, and in so doing, found a new-to-us virus that may have been in these shrews the whole time. But even so, in both manuscripts, Pepper is discussed only as a domestic cat. Despite gathering samples like any student or technician, somehow Pepper was omitted from the paper's authors, and only humans were included in the acknowledgement. Put some respect on fieldwork, I say. Give this cat a PhD. We could use a Dr. Pepper.